Our two guests, Dr. Stephen O'Brien and Dr. Kevin Fenton from NIH and the CDC. Today, our conference is going to be entitled Genes and Disparity in Healthcare Delivery and Behavior. I have to read it because it's so long. What role does each play in susceptibility to HIV? How do we come up with this title? Well, about a year ago, a group of us in the AIDS Institute decided that we should have a conference that touches on obviously a very important question, but also something that crosses disciplines, since we have so many people here from behavioral sciences, basic sciences, and clinical sciences here. And this topic was one that we could all agree upon, which was rare, and also is of an extremely important nature for understanding HIV disease. And as I was thinking about what to say this morning, I was thinking that this topic is something that it really relates back to some old adages of nature versus nurture. And so not knowing too much about that, because I hadn't studied it in quite a while, I thought I'd go to the internet, and I found an interesting article here that some of you here may be familiar with, since it has to deal with psychology. And so if you are already familiar with this thing, excuse me, but it's something that was really new to me. It was an article by Philip Groff and Laura McRae. It was presented at the American Psychology Association, Chicago, in 1998. And the title of their talk was Nature versus Nurture Debate. Nature versus Nurture Debate in 13th Century France. And so they were tracing back the origins of when this debate first began. And they cited work of a poet by the name of Heldris of Cornwall in medieval France, who published a poem around 1250. And this poem was entitled Silence. And it was about a young woman. Her name was Silence. She was brought up as a boy because at that time in medieval France, women were not allowed in any inheritance. So her parents brought her up as a boy, disguised her as a boy. And to make the poem was quite long. But basically her upbringing was extremely successful, and she was found to be superior to all of her male peers in riding, wrestling, weaponry, music, and hunting, although they didn't mention anything about shooting moose. Now, in this poem, there was a personification between two individuals who were discussing the merits of this woman. And it was a vigorous debate between two individuals known as nature and nurture. And each was taking responsibility and credit for her various successes and her character. And so there was heated arguments throughout this poem. And at one point, nature, in one heated moment, nurture in one heated moment turned to nature and said, "Nature, leave my nursling alone, or I will put a curse on you." So this debate of nature versus nurture continues up until today and in this very conference. And it's now reflected even in infectious diseases. And AIDS, which is our modern day curse, is something that we will explore today, the relationship between the genes and the environment. So with that, I'd like to welcome Dr. Steve O'Brien as our first speaker. Dr. O'Brien is the chief of the Laboratory of Genomic Diversity at the National Cancer Institute. And for myself and probably most of you here, he is best recognized for his work on describing the first human gene to influence HIV-1 infection and disease progression, CCR5 delta 32, which I believe he'll be speaking about today. But he has also done a number of, in looking at his bio, a number of very interesting, in his academic career, a number of very interesting projects, which I wasn't familiar with. So he has defined a new species of orangutan, clouded leopard, and elephant. He developed a discipline of conservation genetics, and he's published a popular adventure story book entitled Tears of the Cheetah and Other Tales from the Genetic Frontier. And as a result of all these various accomplishments, including delta 32, Dr. Brian O'Brien was elected to the American Academy of Arts and Sciences, as well as to the Explorers Club and to the Cosmos Club. So I'm very pleased to welcome Dr. O'Brien to our conference today.
Thanks very much, Dr. Chin. It's really a pleasure to come here for several reasons. One is because of the outstanding nature of the work and leadership that has come out of UCLA in AIDS research, epidemiology, surveillance, as well as capacity building. The second is because this is really the place where HIV was originally discovered in American patients as an epidemic spreading among actively gay men in the center of the city, replicating itself in other large cities like the U.S. and like in Washington, D.C. and in New York City. Okay, really in the 25 years since AIDS emerged as a deadly infectious agent among actively gay men, hemophiliacs, and recipients of blood transfusions in these American cities, our country has invested billions, learned quite a bit, but in the end failed to stem what has become the most deadly epidemic in history. In the past generation, this is a photograph from the top of the Washington Monument taken on Columbus Day, 1996, illustrating 40,000 emblems called the AIDS quilt, representing just a small fraction of the American casualties that occurred up at that time of HIV infection. In the past generation, we've watched AIDS spread across our country and the globe and taken excess of 40 million lives, leaving some 25 million people living with HIV infection. Over 30 antiretroviral drugs have been developed, mostly starting out in 1996 and later. And the powerful combination of the triple drug therapy has reduced mortality, at least in the industrialized world, illustrated by this slide showing the progress of mortality starting to slow down when the triple drug therapy was introduced. In addition, the PEPFAR program has gone a long way to deliver therapy to the developing countries, over 2 million people being on heart therapy right now. That said, the incidence of new infections are outpacing by several-fold the rate of new patients treated in the developing world, so that the Red Queen for AIDS is losing ground at the very best. Even so, the best heart therapy the best heart therapy is certainly no cure and is not ideal for everyone. A sizable percentage of heart patients do not suppress a viral load, and also many fail to tolerate the drugs. The virus is never cleared from latent intracellular reservoirs, and the severity of cardiac and lipid metabolism adverse events in many cases are worse than the disease itself. We desperately need better treatments, and some of the most promising have really come from research discoveries of host genetic facilitators of AIDS pathogenesis. Now, in this slide, I just list some recent and quite promising next-generation AIDS treatments based upon blocking of HIV infection, HIV integration, replication, that were invigorated really by basic science discoveries of the roles of gene influences of genes like CD4, CCR5, Delta 32, TRIM5 alpha, APOBEC3G, and other human genes that regulate HIV pathogenesis. Now, the application of new treatment avenues by uncovering the natural genetic impediments to AIDS was really the principal goal, our principal goal in 1980 when our young group first started searching for what we call AIDS restriction genes. Now, an AIDS restriction gene is a human gene with a polymorphic variant that influences the outcome of HIV exposure or infection, and the hope was to discover these to really inform the development of better diagnostics, prevention, avenues for therapy, and even vaccines. Now, our first AIDS restriction gene was CCR5. This is a list of about the 20 AIDS restriction genes that have been developed in the last 20 years or so. Wait a minute now, what's going on here? Okay, yeah. After CCR5, we went on to discover about 20 others using the same cohort-based population genetic analysis. And this is a list of the 20 that were counted in the cohorts that we actually studied. Since in addition to that, several additional labs have mounted searches for AIDS restriction genes in other cohorts, including many in the developing world. My talk today will tell their story together with our own and try to inject some hope and promise, plus a little reality into the image of tears and terror that we know is the most deadly epidemic in historic times. We started, really, as I mentioned, in the early 1980s by collecting good, still connected, amazing. By collecting patients from AIDS cohorts that were at risk for HIV because of their behavior. Those include actively gay men, hemophiliacs, and IV drug users sharing needles in city slums where the prevalence rate was on the order of 80 to 90%. Altogether, with these epidemiological collaborators, including Roger Dettles here at UCLA, we were able to go ahead and collect almost 10,000 people in the cohorts and to transform their blood samples into immortal cell lines using Epstein-Barr virus, which became the source and the currency of DNA that we could use for our population genetic studies. What we're looking for is distortion in allele frequencies or genetic equilibrium in patients in different categories of outcomes associated with HIV. These included specific hypotheses looking for genes that affected resistance to HIV infection. Are there any genes that affect the rate of progression among people who are already HIV positive based upon either a Cox proportional hazard approach with Kaplan-Meiers or a categorical analysis of slow and long progressors and long-term survivors or elite suppressors? In addition, are there genes that influence the AIDS-defining conditions, the type of opportunistic infection or cancer that AIDS patients get? And finally, since 1996, there's been an opportunity to gather phenotypes associated with the heart therapy itself, such as the efficacy of suppressing viral load, the rate of AIDS progression in patients even on heart that have suppressed HIV below levels that are detectable in the serum, and the adverse events, including fat redistribution as well as cardiac outcomes as well as failure to tolerate these. All of these represent specific opportunities for discovering human genes that play some level of a role in determining how well people do. The first AIDS restriction gene, as I mentioned, was the receptor for HIV CCR5. There are now 3 major receptors known for HIV, the CD4 molecule, the CCR5 molecule, and the alpha-4 beta-7 integrin, which is located on gut-associated lymphoid tissue, recently described by the Fauci lab at NIH. When we resequenced the gene for CCR5 in 1996, we discovered a deletion which knocked out the transcription and processing of the CCR5 molecule, such that people with 2 copies had no CCR5 on the surface of their cell. They had 2 copies by inheriting the delta 32 allele from each parent. Those individuals were found to be depleted enormously among people who were HIV positive, but not so much in people that were HIV negative. Because the gene has a low frequency, the frequency of homozygotes and heterozygotes for delta 32 were equally represented among positive and negatives. But the homozygotes, which actually have no doorway for HIV to enter cells, are 20 to 30 times less frequent, with a p-value on the order of 10 to the minus 12. This was the first discovery of a recessive protection against HIV infection in a human population. It also had a dominant effect on what we call AIDS progression. Although homozygotes do not become infected, heterozygotes do. And they show what we call a modest delay in the onset of AIDS based upon the survival curve, where individuals homozygous for the wild type progress at this rate, and individuals heterozygous, because they only have half as much CCR5 on the surface of their T lymphocytes and macrophages, delay the onset of AIDS 2 to 4 years. It's a modest effect, but the statistical significance is real, on the order of 10 to the minus 4 or so. Now, Finally, this delay is associated with a specific AIDS-defining condition, namely non-Hodgkin's B-cell lymphoma, which is reduced by 50% among individuals who are positive for delta 32 heterozygosity versus those that are negative. By contrast, the frequency of developing Kaposi sarcoma, other opportunistic infections, or pneumocystis, common AIDS outcomes, is equivalent in people with this genotype. So we have a gene here that has a recessive effect on infection, a dominant effect on AIDS progression, and a dominant effect on AIDS-defining condition. Other molecules involved in HIV entry into cells were also shown to be involved, including a promoter allele haplotype associated with CCR5, which caused an increase in CCR5 abundance on T cells, causing more rapid progression in this case. This is CCR5 itself. This is a combination of two genes, CCR5 delta 32, and the ligand of CXCR4, a late-stage co-receptor for HIV that is replaces CCR5 in about half the patients in the West that are infected with HIV. Invigorated by these discoveries, we became very careful about listening to AIDS conferences about cellular factors that were being implicated so that we had candidates that we could resequence and look for variants. In the process of the last 2 decades, we've gone ahead and discovered not only CCR5 variants, but variants in other co-receptors and their ligands listed right here that are involved in either prevention of infection, protection against progression, and so forth. And these have been published in pretty good journals and replicated in other cohorts as well. So this was the beginning of our accumulation of genes that have a small but replication showed effect on HIV progression. Beyond CCR5, we also looked at genes in the immune system in our several thousand cohort patients, including individual genes involved in acquired and innate immunity and regulation. I'd like to take a few seconds and just illustrate some of these for a moment so that you'll understand why we believe these are pretty important. HLA is well known as a complex of over 200 genes on chromosome 6, including about 60 of which are involved in presentation of antigens to the helper and the cell-mediated and the humoral immunity of the T lymphocytes. And these particular molecules, such as class 1, which mediate cell-mediated immunity, or class 2, which mediate humoral immunity, were interrogated to see whether or not the high diversity associated with them, first of all, had any influence on HIV progression. And in fact, it does, because people that are heterozygous, if you will, for all 3 HLA loci, regardless of their genotype, progress to AIDS according to the blue line, while those with 1 or 2 homozygous loci actually progress much slower. That is to say, people that are homozygous for all 3 loci progress to AIDS at twice the rate of people who do not, as if diversity itself is important in the population and in the individuals. This was pretty surprising, but it did not explain all of the rapid progression, because only about a quarter of patients that progress rapidly actually are homozygous for all 3 or any of the HLA loci. So we were interested in whether specific HLA alleles are specifically associated with rapid progression or resistance. And what we did then is we did survival analysis on every single one, and what we see on this slide is simply a plotting of the ranked relative hazards of all the HLA alleles that have been tested. And in our laboratory and in several others, 3 specific alleles have been demonstrated to have shown a big effect. One is the B27 and B57, which are good for you and cause a relative hazard significantly below the mean of all the others. And one which is very bad for you, B35, which shows a rapid progression as well. Now, let me just say a word about B35, because the HLA community is actually known for some time that these particular designations of alleles represent not one but several different sequence-specific transcripts and products which have the same serological recognition. So what became interesting about that was to partition, if you will, the B35 effect into the recognition, the functional recognition of B35 itself. And before I show you this, I will just illustrate for you what the Kaplan-Meier curve looks like for the three operative HLA loci, where B57 and B27 are good for you. B35 is bad. As I mentioned, B35 is. If you look at B35 itself, it causes a rapid progression to AIDS in homozygotes or with one with a highly significant effect illustrated right here. Now, B35, as I mentioned, is a member of an abundantly polymorphic HLA-B locus. There's over 200 different transcripts which have sequence that can be organized into a phylogenetic tree, as illustrated right here. And what is interesting is that there's about a dozen different B35 types. And those B35 types, which are blown up in this particular situation right here, actually have slightly different functions, which is to recognize and present 9 amino acid long epitopes to the cytotoxic T cell receptors to recognize the presence of infectious agents in cells. And it turns out that the signature associated with HLA and with B35 consists of a specific amino acid type in position number 2 and position number 9. And if you look at an HLA molecule from the top of a cell, it kind of looks like a bear trap, with the jaws of the bear trap being these alpha helices and the base of it being this beta-pleated sheet. And then inserted in it is this monomer, which contains signature residues in position 2 and 9. And B35 is actually parsed into 2 types. One group that recognizes tyrosine in position number 9, a big molecule, and phenylalanine in position number 2. The second we call PX, which is phenylalanine in position number 2 and anything but tyrosine in position number 9. So we wondered whether or not this recognition would explain or validate the HLA association to rapid progression. And it actually did, as is illustrated in this slide, which is hard to see because the color's a little funny. But let me explain. The gray line is individuals without B35 progressing to AIDS in a Caucasian study. And the blue line is what we call PY, the tyrosine or threonine in position number 9. The red line explains all of the protection. This is PX, and this is in Caucasians. If we go to African Americans, we see the same result, independent replication, even though it turns out that the ones that are PX in Caucasians are actually different alleles from the ones that are PX in African Americans. Again, illustrating that it's the recognition of the epitope that is driving the effect and not the specific alleles and some elusive aspect that's tracking it. This is really a functional demonstration of HLA recognition and presentation associated with a particular outcome of an infectious disease, namely HIV. HIV has a number of, HLA has a number of other roles. One of the most interesting, in addition to presenting epitopes to the T cells, is the interaction with a different armament of the immune system called natural killer cells, which was actually invented in an evolutionary sense to detect cells that had become infected with viral agents that learned how by evolution to shut down HLA class I expression. So what these killer cells do is they survey the body looking for cells that don't have the ubiquitous HLA molecules, and they make the conclusion that these cells are infected with something and they kill them. However, the recognition step involves not only the presentation of the looking for HLA with a KIR, but an HLA-specific interaction indicated right here, so that an uninfected cell expressing both a viral epitope and HLA will be suppressed from killing. However, if a virus actually downregulates the presence of the HLA antigen, what you wind up with is the absence of the HLA ligand, and then the KIR molecule does it. But this interaction is related to the genotype of the HLA allele and the natural killer cell allele, a group of genes on chromosome number 19. So we wondered whether there was specific association of not one, but the two together. And that's actually what we saw when we basically looked for the interaction of the KIR 3DS1 genotype which specifically interacts with a group of HLA molecules called VW4. And when the two of them are together, they actually are very low in the presence of rapid progressors, but very high in people that avoid AIDS for 12 years or longer. This particular gradation, a stepwise categorical test, we call it a multipoint, is an illustration of the influence of the interaction, which geneticists call epistasis, of two separate alleles, which by themselves don't have an effect. Again, an age restriction which is evolved together by two separate alleles on two separate chromosomes, which interact together to cause a functional consequence. There are other genes involved in innate immunity, which we've also discovered and published using the same kinds of methods, including not only the HLA system, but ApoBec, TCSG1, IL-10, suppressor cytokines, and other kinds of genes which have small but detectable effects. In addition to that, there are specific genes that are now being discovered, such as ApoBec and TRIM5, Cullen5, and several others that are involved in regulation of viruses themselves. These are evolutionarily derived restriction factors which were not discovered by the immunologists, but discovered by the HIV people looking for genetic factors that suppress them. And they have interactions with things like cyclophilin and TRIM5 and ApoBec in different kinds of ways, which I won't go into, but I'm sure many of you are familiar with. For example, there happens to be a haplotype of 30 SNPs around the Cullen5 gene, which specifies one of the subunits that's involved in the interaction of cyclophilin and other genes, which show a quite dramatic association with rapid versus slow protection based upon the association analyses like the one I've showed you. Finally, I've said nothing about genes involved in defenses against heart therapy. These are genes that we could test after 1996 when the powerful therapy came up. And they had to do with the epidemiological heterogeneity we know about, that some people actually fail to suppress the virus below detectable. In addition, there are several people that have severe toxicities or side effects of the drugs, as well as several that have more serious side effects, such as lipodystrophy and cardiac complications. So we erected 5 separate hypotheses and simply tested individuals using categorical or survival analysis to see whether or not, first of all, the archival age restriction genes, the ones that we discovered before 1996 cutoff when HAART was introduced, have an effect on heart AIDS progression or on suppression. So what we did is we had a list of about 20 of these, plus a half a dozen that other labs had done. And what we discovered, that we discovered a handful that showed a significant either protection in the case of greens or susceptibility in the cases of reds. These are odds ratios between 0 and 0.5, very modest, or less than 0.5 to 0.1. Several of them are chemokine receptors for CCR5 or CXCI4, and the variants in them have quantitatively been attached to expression, and makes us think that perhaps the presence and availability of these HIV entry portals is important even after suppression, meaning that HIV replication is continuing. This is a close-up survival curve of what happens with CCR5 when the heterozygotes actually protect against AIDS progression after HAART therapy. It's basically highly significant, even though the number of patients is rather modest, about 500 altogether. When we look at RANTES, which is a primary ligand of CCR5, we also see protection associated with alleles that protect in the natural history experiments pre-HAART. In addition to that, if we actually summarize all of the genes that show an effect of the AIDS restriction gene, and we list them over here, these are the significant ones for natural history. The green colors represent genes that are protective and good for you. The red are the ones that are bad for you before HAART. Then what we measure is viral suppression, genes that affect that, genes that affect progression to AIDS after HAART, genes that affect the CD4 slope in suppressors or in all the patients. And what we notice, by and large, is not perfect, but reasonable concordance, where the protected genes for natural history are often protective, as is CCR5, at least this particular situation, after heart, and several of the ones that are bad for you accelerate progression, such as those that diminish Rante's abundance. They're also bad for you after heart. Again, emphasizing that even though suppression leads to undetectable levels, it also can, there's probably a subliminal replication going on, which is limited by these age restriction genes. All in all, there's now a dozen different age restriction genes that affect heart, the pharmacokinetics, if you will, and they're listed on this slide. Okay. I have described for you very rapidly about a half a dozen ARGs that affect HIV infection. That is the transmission of virus from person to person, including both HIV entry portals as well as immune system and antiviral drugs. They're listed on this slide. We've got more data and more genes that affect HIV/AIDS progression. And they're listed on this slide. Again, a number that are involved in HIV entry, a number in innate immunity, and another number in viral suppression. And again, they have different effects. They're located on different chromosomes, and they have different kind of effects, and they've nearly all been published and replicated. Altogether, we believe these 20, even though you hear a lot of noise from the human geneticists about false positives because of multiple tests. Here's why my colleagues and I think that these 20 are okay. First of all, the associations show effects in AIDS patients themselves, not in some experiment in a mouse or in a laboratory setting. Second, our clinical cohorts have very high numbers, between 2,000 and 10,000. The associations yield significantly low p-values even after correction for multiple tests. And nearly all 20 have been replicated in independent cohorts, different populations, and different risk groups. Each age restriction gene was evaluated for independence versus epistatic interactions, as I showed you for HLA and for KIR, and with other genes, giving us a perspective about those which are independent and those which are interactive. Fifth, functional assays have demonstrated that there is diminished or elevated transcription associated with alleles and in the right direction that can be explained by the context of HIV replication using the idea that more virus leads to rapid progression, sort of a tenet of any AIDS pathogenesis study. Finally, plausible functional evidence involving each ARG with HIV. These are not genes, mutations out in the desert in the genome. They're ones that specifically affect HIV and have been demonstrated by other laboratories in non-genetic settings as having a role in HIV replication, spread, and pathogenesis. The reasons why we wanted to go ahead and describe age restriction genes in the first place was principally for these three reasons. There were others, but the major ones are to define rate-limiting steps in progression immune collapse, to identify host factors suitable for therapeutic intervention. I've already shown you a slide which showed a bunch of the things that were stimulated by the discovery of Delta 32. But the third way is that you hear about a lot, particularly in AIDS discussions, which I want to just talk about for 5 minutes, is can we come up with a genotypic signature and quantification that can allow us to weight the likelihood and be predictive about what's going to happen to a patient when he's affected by AIDS, and he has a particular composite of HIV age restriction genes. This would be useful because we don't have a vaccine, we don't have drugs that are perfect, and in fact they're not very cures and they're very toxic. So there's a lot of clinical trials going on. So maybe we can come up with ways in which the clinical trials can take advantage of the genotype of the volunteers so that that can be weighted into interpreting those things. Are you with me? Okay, so let me show you what this requires. In order to come up with a statistic or a number that tells you what an individual should do that's predictive, we simply took the relative hazard of a survival curve and multiplied it together because it's a geometric expansion and came up with what we call the genetic propensity index. It's a predicted relative hazard for an individual's genotype relative to the common wild type. And for multiple genotypes, GPI is the composite RH. You just multiply them together. Okay? Now, if you do that for wild type, well, the wild type is the most common allele. That's always just 1. So you come up with a composite GPI of 1 for relative hazard. But if you take all the protected ones, the ones that are good for you, and you multiply them out, that relative hazard drops to 0.07, which is pretty good. That's almost a 20-fold difference. In the same way with all susceptible, 20-fold, except if you look at ones that are rare, which we call the variants, they offset each other because some are good and some are bad. And it's not much different from one per se. If I take this, so what I've done is I've taken people that are results from all the different tests and combined them together for this GPI. Now, if I plot this, the theory is that people that have all the protective genotypes progress to AIDS like the green line. The people that have all, the rare people that have all the susceptible, like the red line. And the people that have just the simple wild type ones, the ones that were not discovered, are blue. Or all the protected all the different new variants are orange. And the reason that they're similar, again, is because the orange ones has offsetting, as do this. They're canceling each other out. Fair enough? That seems like a good number to have. But is it? Well, it kind of depends on how much of an influence each one of these things have on the overall progression of the population. So we have a couple of parameters which we use to quantify how a factor, an epidemiological factor or a genetic factor, influences AIDS progression, say, or any outcome. One is called relative risk, which is how strong is the effect. For example, if you have R equals 5.0 for a disease that occurs at, say, 1 time in a million, that simply means that if you have this factor, you have a likelihood of getting it of 5 in a million. Not that impressive, but it's a big relative risk. Attributable risk is a different number. This is a statistic where you take the people who are in a particular group, like rapid progressors or people with lung cancer, and you say, how many are in that group because they have this factor? And that gives you a number which is kind of useful. It's usually pretty high, though, because it's not additive. It goes up over 100%. So it tends to be misleading. You can have 3 factors that have attributable risk of 60% for the same disease. HIV has 100% attributable risk for getting AIDS, but there are other factors that contribute to it as well. So we invented a few years ago a new statistic which we wanted to be a yardstick, which went from 0 to 100%, which expressed how much each individual gene, and then in an additive way, how much many and environmental factors contribute to the variance in an epidemic. And it's basically a useful new yardstick to quantify what percentage of the variance is explained by the genetic or environmental factors. So we know where we are, from 0 to 100%, or from one end of the 100 yards to the other end. And it's based upon mutual information theory. It was published a few years ago, and it's computed by a simple 2x2 case control, or the genotypes table, and a simple equation that looks something like this. That's all the math I'm going to do. It's simple to compute, but to illustrate how it works, let me show you, for example, what you get when you do that with a bunch of people that smoke or don't smoke and that do or do not get lung cancer. It's based on a data set that was published a few years ago. And what this shows is that with the non-smokers and the smokers, those that get lung cancer and death, a 2x2 table, when you compute the relative risk, well, it's huge for smoking. It's about 10-fold. When you compute the attributable fraction for smoking, it's 74%. Why is that? Because when you go into a lung cancer ward and you see all these people, 80% of them have been smokers before. But if you go out into the street and you ask the people that smoke, how many of them have had lung cancer if they live long enough, it's much less. It's only about 5 or 10%. So the explained fraction of smoking is 9.7. The difference, almost all who die from lung cancer were smoking, so most of the lung cancer can be attributed. But 98% of smokers never get lung cancer, so the other factors are playing a role. In other words, who gets lung cancer? Thus, lung cancer is only partially explained by smoking, even though we know it's important. Well, do the same thing now for the AIDS restriction gene. The protective arches are these. These are a bunch of things that are good for you. These are a bunch the susceptibles are good for you, and so forth. When you look at the attributable risk, 21% of the patients that are protected are in that category because they contain a favorable genotype or more. The ones that are susceptible, 22%. But look at the explained fraction. Most of them are less than 1%, 2% in this particular case. And the total is like 9.8%, which means we can explain 9.8% of the variance associated with AIDS progression in the Kaplan-Meier curves with the genes that we know about right now. Well, that means that 10 times as much, 90%, we don't know what's going on very well. So, predictions can be tricky, especially ones about the future. What does this say about our ability to take the GPI and hand it over to a clinical trial and say, weight this? Well, it says we're only explaining 10%. So to prove and illustrate my point that we're not where we want to be, we took the GPI of 1,000 people that we knew what their outcome was. And then we plotted their prediction versus what actually happened. And guess what we found? Predicted AIDS-free for survival. This is our intelligent, beautifully calibrated mathematical expression. This is what happened. It looks like a circle. The relative R standard was 0.14, about the same as you'd expect because the explained fraction is only 10%. Sorry, boys, we're not there yet as far as turning this over to the clinical trials. We need to get better. Remember that when you start reading about important genes in any of these diseases. So, summary. Well, the long and the short, only about 10% of the variance in AIDS can be explained by the genes that actually affect survival. Not enough for prime time. The good news is that 9.8% actually isn't bad because it's comparable to smoking for lung cancer, which we all know is important. Another point, genes with relatively modest epidemiological influence are readily discovered and validated with these well-described cohorts. I think there must be other factors out there ready to be discovered, genetic or environmental. So, what's next? Well, this year, a really cool paper came out of Harvard from Abraham Brass and Stephen Elledge, where they described a way in which they wanted to use to identify AIDS restriction gene. And they didn't do what we did, which was genetic association. What they did is they took a HeLa cell, and they transfected it with CD4 and with CXCR4, a receptor, second receptor for HIV. Then they infected the cell with HIV. Then they took silencing RNA for one of the genes on chromosome 1. And then they transfected the cell to silence that gene. Then they infected it with HIV and asked, does it replicate? Does it produce virus? Does it produce antigen in the surface? Then they did it with a second gene and the third. And they kept doing it until they'd gone through all 21,000 of the silencing RNA transcripts there are for the whole complement of human genes. And then they counted up the ones that stopped HIV replication. Altogether, after a couple of filters or so, they came up with 250 genes that they said, these are important for HIV infecting this HeLa cell and for producing it. So all of a sudden, the number of AIDS restriction genes, my whole career, 20 genes, 250 new ones, like that. My buddy Gallo, he says, this is destined to be one of the key HIV papers of the decade, if not longer. I was at a meeting with him the other day, and I said, you never said that about any of my papers. So, anyhow, let's look at the genome with these HDFs, HIV dependency factors. Okay, here they are. Some of them have plausible explanations. They're on most of the chromosomes. The red ones are the ones we discovered with our modest little association studies that I've been describing in the last few minutes. This is just half the genome. The other half genome has another group listed right here. But remember, none of these have been tested in patients themselves. They've all been tested in HeLa cells. And a couple of caveats that you have to understand with this experiment, as elegant as it was every gene that you silence that kills the cell scores as an HDF, okay, because the virus isn't going to replicate it. The second thing is, all the genes in the human genome are not sequentially independent. What does he mean? Our genes are parsed into these groups called paralogous gene families. So if you add a transcript that is meant to silence gene number 8. It's got a lot of first cousins in there, some of which will also be silenced as well. That aspect of it wasn't really straightened out very well. Nonetheless, some of these are going to be important. Probably not all of them, but some of them. Now, we are now in a field where it is possible to look not only at candidate genes, but across the genome in what is called a GWA, or a genome-wide association study. This is a study that does not try to guess what the genes are that are important, but to just saturate the variation by using hundreds of thousands, if not more, single nucleotide polymorphisms. I'm going to tell you about a couple that have happened. And what they kind of do is they basically plot p-values across the genome according to where they exist on the chromosome. This is about 600,000 SNPs, single nucleotide polymorphisms, for a cardiac gene where the MHC was involved, that was published in the New England Journal a few weeks ago. It's basically meant to come up with very high signals that are above the background. Now, David Goldstein, a close colleague of mine at Duke University, and his students published a paper late last year that basically, in August of last year, which was the first genome-wide scan with a large number of SNPs. They used an Illumina platform which interrogated in a microarray 555,000 SNPs. They measured one phenotype, one question, which is CD4 trajectory to less than 350, or viral set point, which is a plateau level that HIV patients achieve after their initial spike. They used a cutoff for multiple corrections called Bonferroni, which simply means you take the p-values and you divide by this number in order to get this number, which is the number below which you don't believe. They identified 3 genes, 2 of which we knew about before, HLA-B57, HLA-C, and then a new one which we didn't know about before. This was hailed as a pretty important, and the first GWA, but it actually wasn't the first GWA, because the first GWA was published in 1996 in the paper that we came out with that was used to discover CCR5 delta 32. It was primitive, it only had 1,000 or so SNPs, but this is the p-value over here, and here's CCR5. This is the way we used to display it. But we were thinking about this before the technology came along. The technology is now here, so where are we? Well, the lab I run is called the Laboratory of Genomic Diversity. And we're completing a GWA now with 2,028 study participants selected from the group of 10,000 I told you about a few minutes ago. We use the Affymetrix platform that interrogates not only single nucleotide polymorphisms, but also assesses a new kind of variation that we're only just learning how to measure called copy number variation. Well, what's that? Well, we are all taught in Genetics 101 that every gene in the genome, we have 2 copies, one for mom and one for dad. And we all were happy with that. But recently we found out, well, it's not quite true. Some people have 1 copy, or 2, or 3, or 4. In fact, 5% of the genome is parsed into these segments of about 5,000 that have copy number variation. So that's another thing we have to worry about, because think about it. If you decrease the amount of a gene, you cut the dosage in half, or if you increase it, you increase it. So things like CCR5 delta 32 or the promoter actually increase and show association. So we need to get at these 2. The Affymetrix chip does that pretty well. Not perfect, but does it pretty well. In addition, about half of the LH-HDF loci are incorporated into complete coverage with a haplotype structure based upon the HapMap. The other half are not. So we designed SNP haplotype across and developed an Illumina chip with 5,000 SNPs to get the other half. So then we combine the 2 together so that we're interrogating not the whole genome, but those 250 HDFs that Steve and Elledge and Abraham Bass described last May. We merge the 2 for 200 HDFs. Okay, what do we see? Well, before I show you that, let me just give you one little last methodology slide. Affymetrix and Illumina are the giants who are competing for arrays. They have similar kinds of chips. The one we used was Affymetrix for certain reasons. There were 6 million little features on this checkerboard, which are little tails of nucleotides that are meant to discover wild type versus variant SNPs. 900,000 SNPs altogether. Each SNP is interrogated by several times. And the copy number variation has as many probes. And that targeted almost 6,000 CNV regions. And then in addition to the ones of the known regions, there were almost 800,000 probes from invariant regions across the genome spaced every 30 or so kilobases apart. And these are meant to be quantified to find CNVs we don't know about. That's pretty cool. It's also very expensive. Okay, a lot of software out there to analyze copy number variation. It's listed right here. I won't go into it in detail except to say the postdocs have developed each one and come up with things that they think works pretty well. I can talk about that more if people want to talk about it, but I don't have time. I will show you this one slide about copy number variation, which I think is kind of cool because it illustrates that we can detect things. If we go through 1,800 patients, we find 9 patients that show long stretches across chromosome 1, 2, 3. These are the 9 patients of copy number variation that goes over whole chromosome. This is a whole chromosome, which is a duplication, whole chromosome here, whole chromosome here. These shaded lines differentiate the chromosomes. These, again, are the 8 people. These are chromosome aneuploidies. Why? Because these came from B-cell lines, and B-cell lines have some level of chromosome aneuploidy that we can detect. This is the Y, or the X chromosome. They all have a deficiency, only one copy. Why? Because they're all boys. They only have one X chromosome. So this is just an illustration to make me feel good that we can detect the copy number variation. Okay, there's a couple of filters that we put through it. These are informatics filters that we use to throw away bad data so we won't be chasing after bad associations, such as probes that don't have complete SNPs because there's something wrong with them. The ones that have polymorphism so low they're useless, less than 1%. Accuracy, we test this in 3 ways. One is we put in a bunch of trios, parents and an offspring, like about 4 of these. And then if they fail to show Mendelian transmission, we throw it out because that's not an interesting result. That's a bad SNP. And then finally, Hardy-Weinberg, we expect the homozygous populations all the SNPs to go around us. When they don't, that's not an interesting population structure. That's a bad SNP. So that's basically what we throw away. There are other methods, which I thought were on the slide, evaluating population structure as well, which we also consider. Now I'm going to show you a few results. All right, this is the first hypothesis we tested. And across the bottom of this slide is the chromosome location. And up here is the log of the p-value. So this is 10 to the minus 6, a little bit less than the threshold of 10 to the minus 7. Most of them fall less than down here in the level of known significance. But many, many SNPs fall up here at a significant level of 10 to the minus 3, 10 to the minus 4, 10 to the minus 5. And a few of them are actually up pretty high. But none of them exceed the transmission, the total level. Now, one of them shows and illustrates what happens when you get an association driven by one SNP in a region where the SNPs are traveling together in what is called linkage disequilibrium. Then a whole bunch of SNPs start shooting up with significant levels at the same time. And that's the kind of result that you want to see because it allows you to go into that data and find out what the gene is that's driving it. That's progression to AIDS, CDC 1987 with a recessive model. Let's look at co-infection. Well, here, we actually set the threshold even higher at 10 to the minus 7. And what we see is about 6 loci on chromosome number 2, on chromosome number 4, on chromosome number 11, and a couple in between that are showing highly significance. These things are definitely interesting because we can go to them and find out what are the genes in there and what's driving it. And if it's not a genotyping error or something like that, then that reaches what we call genome-wide significance. That's kind of cool. So already we've got a half a dozen new markers that have followed this with the genome-wide association. When we look at the question, if you take the patients who get AIDS and they get Kaposi's versus everything else, for the first AIDS-defining condition, what we discover is that we see significance, and there's kind of a cool one here on chromosome 5, which shows a whole bunch of associated loci tracking it as well. Again, surpassing genome-wide significance up here. When we look at any Kaposi sarcoma, a different test, but not independent, well, we see the same thing. Okay? Different group of patients, but overlapping. Again, an internal replication. Now, I'd like to say in heart, when we look for regions that are associated with suppression of HIV, well, there's a couple of regions that look kind of cool. There's one on chromosome 15, and there's another one over here, which doesn't quite reach significance, but we're going to look at it anyway. This is the first shot at a genome-wide association scan using the 2,028 patients that we've talked about. Now, this is the same slide I showed you a second ago, which is progression to AIDS in 1987. 10 to the minus 6 is the threshold. All the AIDS restriction genes, the 20 that I described before, were not detected. Why is that? Why didn't we see them, which was the candidate gene report? Because mixed in to this region, above 10 to the minus 2 to 10 to the minus 6, the ones that we're throwing away due to failure of statistical stringency with multiple tests is a mixture. One group, statistical hiccups. Another group, real restriction genes that don't have a result, a p-value of 10 to the minus 8. How do we get them? Are we throwing away the baby with the bathwater? We're throwing away all 20 of the ones we knew about. So how do we fix this? That's the final point I'm going to make here. One way we do it is not to subtract tests, but to add more of them, and to look for internal replication. So basically, we have a number of tests that we use for infection, for progression, for heart, for disease outcomes, for response to heart, and then finally, principal component analyses for each one, 10 altogether. Altogether, there's something like 172 tests and 10 PCAs. Again, making bone ferroni even more demanding. But we do it because we think that internal tests with different patients, like the Kaposi's I show you, will lead us in the right direction. Then we developed some clever bioinformatics tools that allowed us to look at multiple tests associated with each SNP and groups of SNPs across the genome. This is a two-dimensional ARG array. What it consists of is SNPs arranged according to their position along the chromosome, and then a bunch of tests for infection, a bunch of tests for AIDS progression, and then a bunch of tests for AIDS-defining condition, and then out here for heart. So what we look at is the color, where the p-value is considered as gray if nothing's going on, and then yellow if it's one, orange, red, and black. So the stronger the color, the higher the significance. And what we're looking for is clusters of significance, which indicate replication of multiple infection tests, corroborated with multiple transmission tests, corroborated with multiple opportunistic infection or sequelae tests. Negative control should look like this. This is a region of chromosome 7 where the cystic fibrosis gene is. It has nothing to do with HIV. And as you can see, when we go down across the genome, about a megabase, maybe 100 different SNPs, and all the tests, 10,000 altogether, we see a background that looks like statistical hiccups. When we go to a region which has a known AIDS restriction gene, such as IL-10, the IL-10 locus is here. The proxy SNPs, which are in LD, are the blue ones. And we start to see small but measurable clusters in infection and in AIDS progression. Some of them highly significant as well. That's what we're looking for, kind of high color in this. If we look at the CCR5 region, where there's 3 different AIDS restriction genes, Delta 32, the promoter, and CCR2, which is 18 kb away, well, then it looks quite colorful. We see the 3 genes that are AIDS restriction genes are here, and this is the infection hypothesis associated with CCR5. The level, that's the first slide I showed you. This is progression, which also shows an effect. And then there is considerable linkage to equilibrium, so these regions are tracking the region. You see a lot of color. Compare CCR5, a positive region in color, with cystic fibrosis. We want to see this, not this. This is 10,000 SNP test combinations. We want to go along the genome and look at this. But we still weren't quite satisfied, and I'm going to skip this, because the tests that I just showed you were basically just measuring p-values. I want to know how strong the effect is too, the relative hazard or the relative risk. So in order to do that, we took that array I showed you, put it in 3 dimension, and then we moved it along as if we were at a video arcade your teenagers go to. We drive along the surface of the chromosomes and we look for regions that have color and have elevation. It's like taking a crop duster plane across the US and looking for the Kansas wheat fields versus the Chicago skyline. Let me show you what I mean. This is basically a snapshot, which simply shows the infection tests, the categorical progression, the relative hazard progression. Over here is the sequelae, out here would be the heart. The green ones are good for you. The height depends upon the p-value, and we're looking for regions that have specific high values in multiple tests. Illustrates some real data now is the cystic fibrosis region, where we have a list of SNPs over here going along a chromosome, a list of tests for infection, for progression, for survival analysis, and for AIDS-defining condition. There's not much going on. A few hiccups here. For the IL-10 region, a known restriction gene, we see a signal in infection associated with multiple SNPs. We see an area for AIDS progression, also multiple strips. This looks a little bit like Peoria, Illinois, or Syracuse, New York, compared to the Kansas wheat fields. But when we look at CCR5, using the same thing, we see a huge Chicago or New York City skyline, indicating high LD, multiple replications internally. Can we scale this up like the kids' video arcade? Well, actually, there's a software that the guys use to make the video arcade where you drive along the highway. And we got people to develop it so we could give them a list of tests and all the different kinds of results. And what I'd like to show you now, if you'll just take me about a second or two, a few minutes, I want to show you what this video looks like. And here she goes. Okay. Again, Hardy-Weinberg, a test for good SNPs, infection hypotheses, progression using categorical, progression using hazards analysis, and some of the sequelae, and then out here. But basically, this is a kind of a negative region, and it's real data. Let me just go with it. And what we do is we sit our postdocs in front of these visit screens, and they look for the Chicago skyline, the New York skyline, and compared it. It's not perfect, but basically we're looking at about 10,000 things every unit long. The green ones are good for you and they're going in the right direction. The red ones are bad for you, they're going in the opposite direction. The height, like that one, is a huge p-value. This is the way we think we can find the genes that are nested below the 10 to the minus 7 bone-Ferroni cutoff with multiple hypotheses and different tests. Remember, CCR5 and HLA fired, and each is one of the different ones. We're in the middle of analyzing it right now. We're excited about it. We know that we're going to discover lots of things that need to be filtered, but we believe in the end we're going to come up with other factors that we could not have think of before with both a functional and a statistical approach at the same time. It'll take you 2 hours to watch this thing, so I'll turn it off and go back to my final point. What you saw there largely was a group of data for 2,028 patients. We actually have 8,400. The Office of AIDS Research has given us the money to go ahead and genotype all of these, a $2.5 million project. This will expand us beyond the San Francisco cohort, the MAX, and some of the ones, and add to it Homer, El Soco, Eurochavi, altogether 8,400 patients, which we'll use as replication using not only the monotypic things I showed you where the little things went over 10 to the minus 7, but the ARG highway. as well as ways to get at them connected with the function. These kinds of numbers we're excited about are going to, we think, invigorate the discovery for age restriction genes. So far, we've analyzed 20 that we believe, I've showed you how, they do translate to patient health. The explained fraction is kind of tiny, about 10%, but it's growing. The new tools make the discovery promising with a deluge of data. We believe that GWAS with available cohorts and bioinformatics routine are under analysis today